everybody is now glad to be here with you, and I am particularly happy to welcome you, and
Thank
also
you.
to welcome the people attending this session. This is a webinar, and this webinar is being carried on in let's say in the first international One Health happening from ICBAS. Which is this Faculty of Medicine and Biomedical Sciences in Porto University. So, well, I want to welcome you and hoping that this next hour you'll have approximately, let's say, 40 minutes, the same 40 or 45 minutes you need, and we will not cut your the word, and we will have then time for questions.
Perfect.
Okay. Just a few words to introduce you, and Dr. Amy LeBlanc is the director of the Comparative Oncology Program in the Center for Cancer and Research, National Cancer Institute in the United States, and it is a very big pleasure to having you here. You are coordinating, as far as I know, the Cotc operations and research. Cotc is a kind of consortium on oncological trials in dogs using the pharmaceutical and universities, several universities conjoint effort to develop, let's say, a 10 years or more effort around this issue, which is comparative oncology. Dr. LeBlanc is board certified on veterinary oncology by the American College of Veterinary Surgeons. And she has a vast experience in small animal medicine and surgery and oncology also. So for us, it's very, let's say, it's very exciting to have you here talking about comparative oncology, since at our faculty we recognized the importance of that issue. Porto University has several units, investigation research units, and also faculties working on issues related with oncology. But so far, our project with VetOnconet is the only project, and working and starting this initiative in the comparative area and domain. So we are excited, I mean, to have you here and to listening to your experience and to, let's say, to the information you are going to share with us. So please, the floor is yours.
Thank you. Thank you so much. It is a real pleasure to share what we've learned and to continue to offer, you know, insight, opinion, advice, things that have worked well for us, but things that maybe we could have done differently or better in the future. I think it's really important. We try to learn as much as we can from other colleagues in our community to make us and our efforts continually better every year. So let me share this presentation. Let's pull this up real quick, please.
Take your time.
Just a second. And you have my slides, yes.
You did not send me them. You can send it to me and I open it if you have trouble.
Oh, okay. It was a really big file. I sent it to you yesterday, but it may not have.
Probably you should have send it by WeTransfer. Probably
okay.
The file did not cross.
Okay.
By email.
Well, let's try this. Usually this works, so
Yeah,
we'll try it. Just allow it to share here. Just a second.
Ela está autorizada por ti.
Okay. All right. Let's try to share PowerPoint. Share and get to the.
Yes, it's okay. We are seeing.
Okay.
It's full screen, yeah.
Is this good or no?
No, not the best view because
Oh,
you are
right. Okay.
showing all the slides. Yeah, that's the best view.
Perfect. Okay. Yay. Good. All right. Well, let's jump in then. Okay. So I would say that this audience is probably very familiar with the idea of studying cancer in companion species. For the purposes of today, we'll talk mostly, maybe exclusively about the dog, but there's obviously a lot of reasons to think about other companion species and the value that they may hold for comparative cancer research. So there are lots of reasons to think that this is a great idea. They're a patient-based model, right? So these are not contrived experimentally induced tumors. These are tumors that develop in dogs in the context of their natural lifespan, giving relevant time for tumor and stroma and immune system interactions to grow and co-evolve and mature in a relevant period of time. And as you know, in veterinary medicine, we routinely apply a lot of the clinically used methods for treating human cancer to veterinary patients. So there's a nice amount of clinical overlap with chemotherapy and surgery and radiotherapy techniques, so that when we talk about the patient model of canine cancer for improvement of outcomes for humans, we're all talking the same language about clinical management. And, in a stark contrast to many mouse models that are available, my lab, which I won't really talk too much about today, but my lab is very focused on combating metastatic progression in osteosarcoma. There really aren't too many mouse models that spontaneously and naturally metastasize. So really even thinking about metastatic progression that we see all the time occurring in veterinary patients, that's a unique opportunity, again, and a big advantage over conventional mouse models. And also, I think the last sort of comment I'll make about the value here is that veterinary medical care does not have sort of a standard that must be met before new or experimental treatments can be offered to the patient, to the family, the pet owners. So very routinely in our clinical trials, and we're not alone here, many people who do clinical trials can offer an experimental treatment to a pet owner at the very beginning of their dog's cancer treatment. So it is really helpful to have access to completely untreated tumors because that patient population is less heavily pretreated and more likely to probably respond to novel treatment strategies. So it gives you a better idea of how a new drug might behave eventually. So if there's a couple of papers that have been written, and I'm sure you may be familiar with them, but I'll call them out here. The National Academy of Medicine here in the US did a workshop a few years ago about the landscape of comparative oncology and what are the challenges and the gaps in our knowledge in this field. And this workshop really highlighted what we don't know and what we need to still learn. And I encourage you to read this because it actually serves as a nice roadmap for really getting at that real value of where studying these tumors that we see in companion species can really have value to improving outcomes, not just for dogs, but for people as well. And as you mentioned, our program, we sit within the National Cancer Institute. This office and where we're currently housed is in Bethesda, Maryland, which is very near to the nation's capital in Washington, D.C. So we sit on the main NIH campus with all the other NIH institutes. And we sit within the intramural scientific program. And our parent branch is in molecular imaging. And that I may come back to that later on in the talk because we have some new initiatives that relate to image-based biomarker discovery and artificial intelligence and deep learning applications that we're starting to weave into our dog cancer research efforts. And so just to give you an overview of what my job here is to sort of coordinate these four main components of our program. the main goal that we have is to integrate comparative biology and clinical trial data and experience to advance the pet dog as a patient model for humans. And so we do that in sort of four main ways. I have a lab here at NCI that does, as I mentioned, sort of very focused work on osteosarcoma metastasis biology and drug development. We also, as I'll get into a lot more detail, do a lot of clinical trials with our infrastructure, our clinical trial consortium, the Cotc. We also have extensive collaborations on other topics within the community on immunotherapy, and we have a comparative brain tumor initiative that I'll talk in some detail about. And we also provide support to the extramural NCI community. And that's the component of the National Cancer Institute that gives grants money out to the community, the scientific community. And so some of that grant money pertains to studying cancer in dogs. And so my program provides sort of scientific input and guidance to those grant portfolios and how to really make sure that the strategy of the grants really has a foothold in what is possible to do within veterinary oncology, and to make sure that those studies have the best chance of informing human cancer care. So this, as you mentioned, is the clinical trials, the consortium, the group of veterinary schools in the US and Canada that are united together through an agreement, a contract, like an agreement structure that basically says all of these schools will work together with me at the sort of scientific leadership position to conduct clinical trials of new drugs in pet dogs that seek cancer care at these institutions. And we have chosen to work exclusively within veterinary schools, 24 members. We could have, and sometimes do think about, branching out of the veterinary schools and going into more private practices. There are literally thousands of veterinary specialty hospitals and clinics in the US that take care of dogs that have cancer every year. But what we chose to do is focus within the veterinary academic world, because the types of clinical trials that we do require there to be expertise in laboratory sample processing. And so a lot of community veterinary clinics don't have access to some of the equipment and expertise that is often required. So, you know, a -80 freezer or a centrifuge or a biosafety cabinet or a fume hood, you know, we need those types of things to be present in the place where the trial is being conducted. And we know that veterinary schools have access to those types of equipment and expertise. So we have chosen to keep the consortium membership limited in that way. But we still have lots and lots of people involved in enrolling dogs on these trials. So we don't see it as a limiting factor. Next, I want to talk about a few specific examples of clinical trials that we've done, just to highlight the purpose of the trial and what we learned and why it was important and really critical that the data that we got from these trials was unique and different and important above what was already learned in mouse models. So I have 4 examples here. I have one of an immunotherapy trial that looked at evaluation of IL-12 and IL-2 immunocytokines in dogs with solid tumors. We have also, I won't get into detail, the RGD targeted delivery of TNF alpha, also a published study conducted in dogs with solid tumors. We have an oncolytic virotherapy trial that we did about 3 or 4 years ago. And then we did a small molecule trial of a group of 3 novel topoisomerase 1 inhibitors. So I'll walk through each of these, 3 of the 4 in some detail. So the first one is COTC 007B, which is just the trial label number. And as I mentioned, it was a study of 3 distinct topoisomerase 1 drugs, small molecules that were developed here at the NCI in partnership with Purdue University in Indiana. And basically the question that was posed was, how are these agents different from each other? We knew structurally they were slightly different, as I pointed out here. But in the preclinical mouse data set, there really wasn't a whole lot of information to say which one was better or worse than the other. And so we created a clinical trial in dogs to very closely examine the relationship of pharmacokinetics and drug exposure to biomarkers of drug effect at the tumor tissue level. And so what we learned actually was that one of these 3 agents, the NSC 706744 in the upper right-hand corner, was distinctly different than the other 2 in terms of its biologic activity. And we were able to, through serial tumor tissue collections, to look very closely at a variety of different biomarkers of drug response and drug activity to try to link the exposure of the drug in the bloodstream to target modulation at the tumor tissue level. And we actually published a separate paper specifically on the biomarker induction of gamma H2AX in tissue samples that were collected from these patients. And that strategy was then applied to the human trials of these agents in the phase 1 clinical trials that are actually still ongoing. And so before I leave this topic, I would say that one of the really most important things about this trial that really called out the value of using the dog to study these drugs was that we randomized across all 3 of these agents simultaneously. We didn't just study one and then study the next one and study the third one. We did them all simultaneously. And we used, the picture here is of a dog with lymphoma. So they have bulky lymph node disease that's pretty easy to biopsy and take serial tumor tissue sections from. And that enabled the biomarker part of this trial to be very, very biologically rich. And so these are things that you can't do really, at least in the US, you can't do this kind of work in humans to that level. You cannot take multiple different tumor biopsies at 1 hour, 4 hours, 6 hours post-drug treatment. There's no way that you could probably do that level of sampling. And we had so much access to these samples that we were able to look at a variety of biomarkers to try and determine which drug was actually modulating its target and which of these 3 agents maybe had a different mechanism of action. So this was a really pivotal trial. It took 2 years to do this work. It was a long clinical trial. It had 85 dogs in it. But it was definitely an example of where working in a multi-center space across our consortium was highly impactful. The next trial that I want to talk about is this COTC 010, which was an immunocytokine trial. And so the story here is that the company that created these immunocytokines was very interested in, so this was about 10 years ago, and immunotherapy now is an incredibly hot topic. There's lots of people working on immunotherapy for cancer, and the field's pretty crowded with new and exciting immunotherapy approaches. But when we did this trial, immunotherapy was not really as widely talked about as it is currently. So the dogs in this trial were enrolled in like 2007, 2008, 2009. So it was quite a while ago. And the company that had developed these agents was not sure whether they wanted to actually continue developing them. They were at a moment where they were thinking, should we abandon the clinical development of these agents? And they needed a data set to really get enthusiasm around continuing to work on these drugs for human use. And we did a clinical trial of these agents, I'm showing you here just one of the two immunocytokines were published. There was a bifunctional IL-12, IL-2 linked immunocytokine and one that just had the IL-12. This is a fully human IgG1 targeted agent that had IL-12 attached to it. So the idea is that you would have an IL-12 induced effect, but without the systemic side effects of cytokine therapy, which are significant. And so we did the study in dogs, and when we developed the data set that's published here, the company actually had a renewed enthusiasm to continue moving the agents forward in people. And so just a couple of years ago, in 2018, actually, the first in human phase 1 work of that same agent, NHS-IL-12, was conducted here at the NCI in a phase 1 setting. And I think my slide might have this in here, but okay. Yeah, so the key point here is that when the dog data was received from our clinical trial, the agent was reprioritized. And when they did the human work with the same drug, the canine data, the dog data, was almost identical to the human data. So a very similar adverse event profile, a very similar pharmacodynamic readout. So the responding immunocytokines that were released into the dog serum were very similar to the cytokine profiles that were obtained from human patients. And the dose that was the maximally tolerated dose in the dog was very similar to the human dose. And so it's a great example of where there's proof that dogs from an immunological standpoint can be studied with high relevance to people in terms of drug development activities. The last trial I'll walk you through just quickly is an oncolytic virotherapy trial. And this was a study of an oncolytic vesicular stomatitis virus that was engineered to express the human or canine interferon beta gene, and also had the NIS imaging reporter, so the sodium iodide symporter, was also engineered into this virus. So theoretically, you could image viral trafficking after a systemic dose. And the short story here is that we identified an efficacy signal in dogs with high-grade peripheral T-cell lymphoma. Both of the dogs in this trial that had objective responses to a single dose of virus were boxer dogs, which we know to be predisposed to a very aggressive form of T-cell lymphoma. They have low MHC class II expression. They're CD4 and CD45 positive on flow cytometry. And these two dogs had a very, very similar response. Have a separate investigational new drug application for this virus in humans with peripheral T-cell lymphoma because of our dog data. And the Food and Drug Administration here in the US took the dog data and was very, very pleased to see what we had learned in dogs and used that to optimize the monitoring plan for the human patients that would be receiving this virus. So just 3 good examples of where there was direct improvement and excitement and enthusiasm around having not just mouse data that we all sort of see as a huge component of drug development and discovery, but also having a bit of dog data to help provide a more clear picture of how the drugs will behave eventually in a human patient. The other thing I want to mention in the terms of clinical trials is that one thing that we know and we believe to be very important is harmonized terminology. So when we talk about side effects of cancer drugs and really any drug that's considered investigational, when we do clinical trials in dogs and cats, it's important that how I describe a side effect or an adverse event is something that you will be able to understand in the context of your clinical trial. So everything is harmonized. So we recently, just earlier this year, revised the common terminology criteria for adverse event reporting. And This paper is an open access paper. I encourage you to look at it if you're involved in veterinary trials, because it really improves the terminology and the language around side effects. And it's not really just for oncology clinical trials. It could be used for any type of clinical trial for arthritis or heart disease or skin or kidney disease. It's a really nice document that helps sort of everybody be on the same page, so to speak. So that, I think, is also a really important part of any sort of multi-center initiative. And then another sort of paper that I think would be important for just a good sort of overview is this Nature Reviews paper that we wrote earlier this year, actually last year, that calls out more examples than I have time to talk about today, in terms of really showing specific examples of clinical trials that have been carried out in dogs that have value for humans. This paper goes into a lot more detail, so if you're interested in learning more about other clinical trial concepts, give this one a read. And this is just an overview of all the different clinical trials that my program here at the NCI has conducted since it started 15 years ago or so. I came here 7 years ago, so right around COTC 20. So about halfway down this list is when I started to get involved here in my position as director. And what I think you can see from this is that there's a whole variety of different types of trials that have been conducted over the years. There's small molecule trials, there's immunotherapy trials, and more, I think you'll see at the bottom of this list, there's a real shift towards immunotherapy. And that was really not the case 10, 12, 15 years ago, right? We weren't really thinking much about immunotherapy, especially now it's like all anybody wants to talk about is immunotherapy. So I think the dog really has an opportunity to participate in that space. So I want to talk a little bit about how doing these clinical trials in dogs has really helped specifically our efforts in osteosarcoma. And there are three sort of topics here. One is drug development. So obviously our goal is to improve that process of making it easier, faster, better to get good cancer drugs for human patients with osteosarcoma. We're also interested more biologically at identifying if there are subtypes of osteosarcoma that have been identified in people. Do those same subtypes exist in dogs? And more importantly, I think from a molecular and biological standpoint, trying to understand the comparative origins of osteosarcoma across species. So how does the disease form in the dog, and how do we think it forms in people? And are those early events, are they shared? And if so, how and why does the disease develop in these two species, and how can we learn more about that process? So I'm going to share with you some of our initiatives here too. So a lot of you, I'm sure, are very familiar with osteosarcoma in dogs and maybe also in people. This is a really common disease in the US, so probably at least 10,000. I would say that that's probably a low estimate for how common the disease is in dogs in the US every year. So a lot more common than it is in people. In pediatric and adult or young adult or adolescent patients, there's probably only about 1,000 new cases in the US, which is why it's really important for us to have global collaborations, because it is such a rare disease on the human side. Dogs and humans share a lot of features. They have very similar imaging, very similar pathology, and very similar clinical characteristics. In that metastasis is the reason why dogs and humans succumb. That's the reason for death in both sets of patients. And more, this disease in the dog is being held up as a really nice patient model. In the paper, the Nature paper that I mentioned a second ago, there's this figure that talks about the shared molecular landscape. And more as we learn how to work with genomic data from dogs with cancer, we start to uncover these similarities. And I've called out a few. This is not an exhaustive list, But there are some similarities in this figure that I think are really interesting from a biology standpoint. One that I'll specifically call out here is MYC. MYC gain appears to be potentially prognostic negatively for humans, and copy number changes of 6 to 8-fold above baseline appear to be prognostic potentially for human patients. We are very interested in understanding the role of MYC amplification in dogs and trying to understand whether it has similar clinical implications. So we've been doing clinical trials in dogs with osteosarcoma for several years now. And doing those trials has allowed us to create a biospecimen bank of tumor samples from these dogs. We have about 450 canine patients that we've worked with over the last really 4 to 5 years, really. And when we enroll them on a clinical trial and we harvest tumor tissue from them, and then we have the ability to then watch to see what happens to that dog as it goes through the clinical trial, if it responds to treatment, if it develops metastasis and when, how long it lives, et cetera. So the samples that we collect have clinical annotation that's really valuable, because then it allows us to go back and say, okay, if we have a group of dogs that lived a really long time, let's go back and look at their tumors and try to understand whether there were genomic features that set them apart from, say, another group of dogs on this trial that didn't live very, very long at all. So we worked with funding from the NCI, who funds our program, but also from the Morris Animal Foundation, who provided the funding for the clinical trials that I'm talking about here. There are 3 arms to this trial. The first 2 arms have been published in Clinical Cancer Research earlier this year. The question we asked in the first 2 arms of the trial was basically this: if you take a group of dogs with osteosarcoma and you give them a standard of care treatment, so that is defined as amputation of their affected limb, and then you give them 4 treatments of carboplatin, chemotherapy, how is that group different than if you give them standard of care, but then you follow that with 4 cycles? So each cycle is 28 days, or about a month, 4 months of oral rapamycin treatment. Rapamycin is sirolimus, it's an mTOR inhibitor. And so that was really the question: is there improvement to be gained by adding sirolimus rapamycin to the standard of care regimen? Unfortunately, we did not see an improvement in the outcome with that. And I think some of the reasons, I don't have time to get into the details, but I think some of the reasons had to do with the rapamycin exposures, and the pharmacokinetics were so widely variable across the dogs that received it, that I think some dogs got a therapeutic exposure to rapamycin, but I think others did not. It was a pretty well-tolerated regimen, so I think the compliance of the pet owner was good, but I think that there was enough variability in the pharmacokinetics that we didn't have enough dogs reaching a threshold exposure to make that a positive trial. I think that's one reason maybe why that trial failed, but certainly pull that manuscript and read it. We went into some more detail about that. The third arm, excuse me, the third arm of this trial is still ongoing. It's the COTC 026 trial, which is where we've again taken the standard of care and added a form of immunotherapy, a HER2/neu targeted listeria vaccine, under the guidance of Dr. Nicola Mason, who's a veterinary oncologist and internist at the University of Pennsylvania. So that trial is ongoing. The data will actually be mature next month. So I'm really excited. I think early 2022, we'll have results to share with the community. The reason why that's really important is because that agent, that identical listeria targeted vaccine, is being studied in human osteosarcoma patients under the Children's Oncology Group. And so we're going to have the opportunity to see if the dog data, again, matches the human data through equivalent clinical trial experiences. So this is just the paper that I mentioned where we did not see an improvement in outcomes with adjuvant rapamycin treatment. So certainly feel free to take a look at that if you're interested in more of the details. Okay, so the next, just a little bit more about some of the work that we're doing with genomics and tumor heterogeneity in osteosarcoma. Um, we have launched an initiative called Dog Squared, which is deciphering or decoding the osteosarcoma genome of the dog. And we're using, again, that group of tumor specimens that were harvested from these trials to ask questions about the genomic landscape and look at subtypes, genomic subtypes that we can define through whole genome, whole exome, and RNA sequencing platforms, and really try to define prognostic signatures and subtypes that can be related to what is happening on the human side. So we have, there are lots of good human osteosarcoma data sets that are publicly available. One of them is the Nci Target Data set, which was developed here at NCI. I think most of those samples have clinical outcomes, so that will allow us to harmonize maybe some of these subtypes that we identify from dogs that were really elite responders that lived a really long time, so they're at the very tail of this Kaplan-Meier curve, versus those that were really early failures. So those were dogs that developed metastatic disease very early in the course of their disease. So this is ongoing. We literally just launched this initiative last year, so more information will be forthcoming from this. And our idea here is, again, to pick out from this group of dogs who we can prognosticate for. We can say all of them sort of look similar, but they have very different outcomes, and we want to understand what genomic factors might be responsible for that. One of the ways that we've tried to address this is to develop good computational tools to work on how to interpret data from canine cancer genomes. So canine cancer genomics is a very small group of people. There are not very many people who have wide expertise in working with the canine genome. Its annotation is not very deep. It's not as well mapped and annotated as human or mouse. So over the last few years, we've worked with a group of bioinformatics experts to create a computational pipeline. So this paper just came out a few days ago. So please go ahead, again, open access, take a look and see. This is basically what the paper talks about, which is a combined experimental and bioinformatics pipeline. We started with canine cancer tissues that were isolated from 5 different tumor types. We did RNA sequencing and created gene expression modules that were unique to the tumor types, but also shared by each tumor type. And we're then able to integrate the data with human data to really see where there's canine-human overlap. Another thing that we're working on is nanostring profiling. So for those of you who are familiar with nanostring profiling, transcriptional profiling, it's a very user-friendly tool to look at gene sets that are specifically curated, and in this case, it's an immuno-oncology panel. Nanostring created a canine panel for questions relating to the immune landscape. It's got 800 genes across 47 different annotated pathways, and we are really excited about this technology and have incorporated it into the Dog Squared project with a specific aim at looking at immuno-oncology similarities and differences between dogs and humans. So the idea here is to really incorporate all of this into a self-perpetuating mechanism where we can identify that landscape genomically, we can integrate with humans. Hopefully that will allow us to identify some new druggable targets and pathways. We can take those back to the lab. So my lab does a fair bit of mouse modeling with dog in mouse and human in mouse xenografts and genetically engineered models to really prioritize new drugs and new approaches that we can then bring back to the dog and say, let's do new future clinical trials in dogs to see if these agents really work in dogs, and offer that back to the pediatric and adolescent human oncology community so they can have new opportunities to treat human patients. Finally, I just want to spend a few minutes on some of the brain tumor research that we're conducting. This is all collaborations with others within the community. Brain tumors are really pretty rare. but equally devastating in dogs and humans. I think all of us recognize the shortcomings of rodent models. And we were wondering, thinking about the gaps in brain tumor research, whether dogs could fill some of these gaps, and how could our program here at NCI facilitate an improvement in research and exchange of ideas. So we held a meeting and talked about some of the real opportunities and strengths that we could continue to work on. This was back in 2015. And that led to, again, a white paper sort of talking about what those opportunities could look like and how we could work together. And we created working groups, 5 different working groups in the sort of bottom part of this figure that had very focused questions around pathology, molecular markers, drug development, tumor immunology, biology, et cetera. So we created a list of projects that we thought were low-hanging, sort of easy things that we could enter into together to make some important new discoveries and new information and share that with the community. So I'm going to just talk about a couple of them because I know I'm running a little short on time. The first one was a comparative glioma pathology board. So basically what this looked like was a group of veterinary and physician pathologists working together to look at canine glioma tumors under the microscope and talking to each other about where they were similar and where they were different, and how physicians would view canine tumors in context of what they see in human patients, and where there could be opportunities to study dogs with glioma to improve new treatments for humans. And what that led to is this cancer cell paper that really harmonized both the pathology and the histopath and the those sort of tissue features with genomics to really get at the highlight of this was that glioma that develops naturally in dogs looks maybe more like pediatric glioma, not adult human glioma. And that becomes really important because on the human side, pediatric and adult gliomas are really different. They have a very different molecular landscape. They respond very differently to therapy, and they have very different outcomes clinically. And so understanding where the dog fits in that spectrum was a major finding. And so we helped initiate sort of the path towards that finding with this work. We also published a harmonized classification scheme for the dog. And so that really has informed many additional brain tumor trials and brain tumor research efforts in the community in the last 3 to 4 years. I'm just going to skip over this. I'm happy to make these slides available too if anyone's interested in looking at them later. The other thing, just sort of mentioning that work then spurred the NIH to fund additional work in dogs with brain tumors and other types of tumors that I've listed here. So this is a list of people who received NIH funding to characterize genomically a whole different list of canine tumors to help improve our knowledge of that. And that also led to publication of harmonized MRI imaging standards for brain tumor trials, and 2 U01 grants that were funded back in 2017 that are still ongoing. 2 of these 5 clinical trials are brain tumor trials. And the work that's coming out of these clinical trials is directly informing human clinical trials with the same therapeutic approaches. So we felt like this was an endeavor, highly collaborative, highly interdisciplinary, that really had a lot of impact on the brain tumor research community in the last 3 to 4 years. The last thing I want to tell you about that you may not know about yet is that the National Cancer Institute has created an integrated canine data commons. So this is a public repository of canine cancer data that lives in a cloud-based repository. And it currently contains a lot of different data types that span imaging data, genetic or genomic transcriptomic data, clinical data, and eventually it will also house immunological data. And this resource allows you, anyone from the cancer community, to analyze, but also more importantly, work with tools that live in this cloud resource. And this is the how you access it. So the web and API interfaces are here. And you can go to this and ask and query these data sets if you're interested in canine melanoma or canine glioma or osteosarcoma. We have deposited some of our data into this commons, and it is there for people to work with it and then use the tools that are there in the cloud to integrate it with human data sets that are also part of this larger cancer resource repository that the NIH has created. So again, this is just one part of a larger cancer research commons. Some of you may be familiar with this already or have used it for your own purposes. It's a really exciting opportunity, and it was really just developed about 3 years ago. And more people are depositing data into the commons. So if you have questions, I can connect you with the people who allow access and provide troubleshooting. So please let me know if you're interested in being connected to this. So, and just an example, these are just sort of high-level questions. If you wanted to know more about gliomas, are there mutations that are specific to certain dog breeds that get glioma? Are those mutations also present in humans? This is an example of where this data commons would facilitate those types of questions. So I'm going to stop there. I would be happy to stay on the line or monitor the chat for questions. We have a really wonderful group of people that work with me here at the NCI, but also from our community and from around the world, really. So we're so pleased to be able to share with you some of our work, and I'm happy to take any questions that you might have.
So thank you, Amy, for your fantastic presentation. And I'm sure you're going to have some questions from the ground. Okay. And while they are coming on, I would like to ask you, from your experience, is there differences among dog breeds? Because we find so many differences. And among, let's say, the sex of let's say, the dogs being neutered or not, I mean. So do you have some income from that?
So I think that that's an incredibly important question. One of the things that we're looking at very closely in the osteosarcoma data set that we have is influence of breed. In the US, you probably know this, but the vast majority of pet dogs in this country are spayed or neutered at a very young age.
Yes.
But we just don't, this is a major limitation of our work, in that landscape of intact male and female dogs is missing from a lot of our data. We don't have the opportunity to look distinctly at neutered or spayed versus intact female and male dogs to understand influences of sex hormones on tumor development and tumor predisposition. So that is a shortcoming of what we're doing. But what I can say is that at this point in time, we haven't seen really major differences in breed and outcome. What we are seeing, though, is that there are some breeds of dogs that might have higher incidences of certain genomic changes. So like Rottweilers in our set appear to have a higher incidence of loss of CDK, cyclin-dependent kinase N2A or N2B. That's been previously published. And so we wonder if Rottweilers with osteosarcoma may have defective cell cycle control pathways, and that might contribute to their development of osteosarcoma. We are also interested in looking at tumor location and the influence of certain breeds of dogs and their stature. If they're really, really big versus smaller, does that influence whether they get it in their front leg or hind leg? We have some ability to look at that. But I don't think that we have seen significant differences in breed and outcomes. But we're still working, to really define some of those relationships.
But that's really an interesting question, because if you didn't have, let's say, we can work with cohorts, more let's say, more ample cohorts than remain limited to a certain breed or so.
Yeah.
And, okay, we have already some questions from the ground, and one concerns the animal model, the dogs being animal models, and our colleague, she asks whether let's say, we can use information from the human side to let's say to address cancer in cats or dogs. Let's say, and the what she mentions is the breast cancer.
Yeah. So this is again an area where our program has not been able to really leverage the tumor type breast cancer, because we don't see a lot of it in our patient population. And I would say that specific to me and the way that we work with veterinary schools, I would tell you they don't see a lot of patients with breast cancer coming to that clinic. Okay. If we chose to work within community hospitals where routine veterinary care was taking place, we would probably see and have access to more patients with breast cancer. But we just simply don't have access to the patients. Okay. And I think it's, again, a shortcoming, but, you know, I'm not certainly the only person in the US working on comparative oncology. So there are definitely other people who would maybe have access to better patient populations. And actually, one person who's doing a really wonderful job with that is Karen Serenmo at the University of Pennsylvania. She has a shelter dog population. So these are stray homeless animals that are coming to the animal shelters in her area. And a lot of them are intact. They've had puppies. They weren't spayed as young animals. And they get breast cancer just as a consequence of having that intact hormonal life. And she is working very hard to apply new avenues to help them from what can be learned from humans. So I would say I wish I had more to contribute to that, but it's just not a patient population that I have access to currently.
Okay, we have another question here regarding osteosarcoma. And the question is whether you are looking for another kind of immunotherapy besides the listeria vaccine approach.
So we are, actually. We are, if I might go back to this slide, if you permit me just a moment, can go back. Actually, I'll stop there. There are 3 immunotherapies that we are currently working on. One of them is a project with an oat bran derived beta glucan molecule that appears to.
You're not seeing, Amy.
Oh, hold on. Let me
stop presentation and shift again.
Yeah, let me see if I can.
Yes, now we are seeing it.
Let me go back. I'll go back to this slide. Hold, please.
Right. No problem.
Let's see. It's going to be easier for me to call it out. Here we go. Okay.
Okay.
There we go. So let's do this. Slideshow. Like that. Maybe? Yes. Okay. So at the bottom, so COTC 029A, 030, and 031 are all 3 novel immunotherapy concepts that we are working on right now. One of them, 029A, is a oat bran derived immunotherapy that we're working with investigators from Case Western Reserve University that appears to modulate macrophage behavior in animal models of osteosarcoma. And so we are working on a pilot study to study the impact of that drug in dogs with osteosarcoma. We also have another trial that we're helping. The University of California at Davis has an inhaled recombinant human IL-15 trial. So dogs are receiving inhaled IL-15 after they have the leg amputation surgery, and then after they finish 2 weeks of inhaled IL-15 therapy, they go on to have chemotherapy. And then the last one are checkpoint antibodies. So those are trials that are being conducted here at NCI In 2022, probably we'll be ready to open them. We've developed antibodies against these checkpoint molecules, and we'll be looking at active anti-CTLA-4 and PD-L1 based monoclonal antibodies in osteosarcoma. Actually, a lot of different canine tumors.
Okay, I have here another question regarding brain tumors. And I think the question relates with the clinical signs in humans, and it regards how to assess the alterations of let's say clinical signs which are somehow easy to collect in humans, like memory loss. Let's say what are the physiological or the criteria or other type of criteria you use to compare the outcomes of
So just one moment. I'm going to be like a while. Sorry. No, I'm sorry. It's going to interrupt it. So brain tumors in dogs are really, really difficult, I think, for us to identify because of just the very nature of how dogs interact with us as pets. It is very difficult to identify them early. And when we did our glioma work, we found that almost every single dog, we had 200 dogs in that glioma project. Almost all of them had absolutely massive tumors involving the frontal lobe. The frontal lobe was the most common place for those tumors to grow in that study. And seizures were the #1 presenting complaint. And so unfortunately, we didn't have any means to identify them early in the course of their disease. I think pet owners are the best source of information because they can tell us something's not right, their mentation's off, their activity is low, or they have other sort of subtle signs of not feeling well before they actually develop seizures, which is a very late stage of involvement with the brain. There are some good tools to identify neurologic status, and I want to say that those have been updated recently. And in our the Veterinary Cooperative Oncology Group document that I showed about adverse events, we recently expanded that document to include much more information about neurologic status. So that might be helpful to get at that question. But I think, unfortunately, we don't see them when they're very early because dogs, don't read the newspaper or drive a car. So
But then I think we have come short of questions, but I have still a few for you. And one is exactly regarding the difficulty we might have in detecting tumors in dogs or in cats. And the question is whether you think it would be interesting to follow, let's say, some kind of identifying some kind of biomarkers which can be used as, let's say, to detect, no, sorry, I'm missing the word, to screening, to do surveys, to screen, to let's say, to do population surveys to these animals at certain ages, to detect them like as subclinical or even clinical, but not let's say perceived by the owners. This is an interesting lead or not? And if you do, how are you addressing this issue?
So actually, I'm glad you brought that up because let me show you. Sorry, I'm going to go fast. This slide and this. So this paper that we just literally got out last week talks about the transcriptional profiling of tumor tissue, which identified biomarker candidates that of a certain component of the genes that were differentially expressed in tumor versus normal tissue a subset of them are secreted proteins that you can detect in the peripheral blood. And so this paper actually goes into some detail on that very point that you just raised, and identifies across all these 5 different tumor types that we studied: pulmonary carcinoma, lung cancer, melanoma, osteosarcoma, and then the T and B cell predominant forms of lymphoma. The model activation profiles and then the transcriptional profiles were mined. I don't have this figure to show you exactly right now, but it's in this paper where there were genes that become biomarker genes that have that code for secreted proteins that could be picked up in peripheral blood. And we have a plan, actually, a proteomic data set that matches this data set that we're currently working on the manuscript for that. So that should be out next year. So hopefully that will help with your question about early detection and screening methods.
We were looking forward to see that work, because in fact, I think to conduct populational studies, we are, let's say, treating data in our system, and we have concluded that data coming from pathological laboratories, they have different frequencies than the ones coming from the veterinary clinicals and hospitals. So the simple issue of cancer incidence in dogs or in cats, I think it needs to be, let's say, to have a bigger and more cautionary and more critical insight. I don't know what is your opinion on this issue.
I agree. I agree. I think that the human field is very much moving in that direction of non-invasive screening tests and monitoring methods. And especially when you think about, like, I spend a lot of time thinking about osteosarcoma patients, and they're pediatric, they're adolescent young adults. And oftentimes it's not, and really any cancer patient, you shouldn't, always be thinking that they have to have invasive procedures to identify what's going on. We really should be looking at more non-invasive circulating biomarkers that can be validated, that improve and optimize the management of patients. So I agree. And so this paper opens that door, and then the subsequent work that will follow will show that in more detail.
And last question, because so far we have not been losing participants, so it is. But so I have, let's say, a last question before closing, and this question relates and is just a curiosity. In our database, we have cats and dogs, and we found among a few thousand samples that cats tumors, they tend to be with higher malignancy in frequency. Let's say cats, they have apparently they have smaller incidence, but the tumors seems to be with higher malignancy. Do you have a comment on this?
I completely agree with that. And the reason I agree with it is because I was trained clinically before I came here to do research. I spent 10 years treating a variety of patients at the University of Tennessee's Veterinary School. And we very rarely ever saw cats with benign tumors.
Okay.
And I think that there's some old data now, which needs to be updated. So your point is a good example of something that we need to put more focus on. Cats are an underserved species. We don't know nearly as much about them in their care. There's not as much interest in them, maybe because, at least in the US, they're not part of the routine sort of plan for drug testing. Dogs are almost always used for drug testing and experimentation, but cats are very rarely used. So there's not a lot known about them. But I remember an old data set that suggested that tumors in cats were 7 times more likely to be malignant than benign. So I completely agree with you.
And that was, I think you partially respond to my last question. Really, this is the last. You don't use cats in your trials, and that is for some particular reason, or you think cats are not a good model for you?
No, we would actually love to, and it is actually potentially coming. One of our COTC investigators Timothy Fan at the University of Illinois has a study in cats with squamous cell carcinoma of the oral cavity.
Very frequent in Portugal.
Yep. And he's studying a new treatment for humans using cats as a model. And we may actually be helping him with a larger scale trial. He's doing some pilot work in his clinic. And if he, and so we already told him we would help with a national trial for him. So we would love to get more involved with good concepts for cats. Yes.
Well, and I am closing now. This is now 1 hour. I thank you very much for your very interesting and informative exposition. It was really very interesting, and we knew already that the work your group and you were doing, it was very interesting, but now we know that for sure that you are really going very in front of us, and we would like to keep in touch, and let's say if we have some talks from here, we can perhaps start better communication and develop communication tools in order to help us to so let's say to go deeper in the problems we both will have and we are experiencing in our some of our participants. And I thank them for being here. They are doctors. They are research community from human side, and I was very glad to have them aboard in this presentation, because in Portugal we are still starting this comparative issue, and it is very important to have it. And I hope from now on we can go deeper in these issues.
I agree. I will say just one last thing in the chat about osteosarcoma. There's a comment about a Brazilian study looking at agonists for TLR2 and 4 and interferon. So that's really exciting. Please check out the integrated data commons that I mentioned at the end of the talk. There's a data set in there that you should have access to. It's our standard of care data set. So it allows you as an osteosarcoma investigator in dogs to compare your treatment to our standard of care arm as a comparator. The one thing is that for it to be a valid comparison, you'd have to have the same sort of monitoring schedule of how often the dogs were checked and monitored clinically for it to be, a good comparison. But please look at that data set, because I think it's exactly what would be needed to help improve study designs for around the world. So there's 126 dogs in that data set. So please check it out.
Thank you. And you still left one issue that for the next talk, which was the imaging issues.
Yeah.
Which you did not address, but it perhaps later in the next talk we can, because it's in fact a very, very interesting theme, and it's very important issue. Yeah,
I totally agree. Well, till next time.
Amy, thank you very much for your presentation, and see you next time. Bye-bye.
Thank you so much. Take care, everyone.
Bye-bye.
Bye.